All right, on this slide, you have DNA structure and replication. This is where the DNA is perpetuating its sequence. Okay, so this is where it's going to split apart, but it's going to continue having the sequence carried on in the parental strand, making the new daughter strand so that this DNA molecule is exactly the same thing as this molecule is over here. So the two parental strands make sure that the two daughter strands match up and that they make a copy, complete duplicate copy of each other. All right. So in this way, they are, again, semi-conservative in their replication. All right. Moving on to the next slide here. Again, DNA replication requires the two parental strands to separate. So you've got to unwind them. You've got to break the bonds. And then new daughter strands come in to make the two new molecules. So only a small stretch of DNA is separated into single strands at any one moment. So you're going to have these little bubbles. It doesn't start like a zipper. It doesn't start at one end and go zip all the way up to the other end and making new daughter strands. And then you got two total new DNA molecules. It starts in little bubbles. So you got a little bubble here, a little bubble there, a little bubble here, a little bubble here, all along this DNA strand. And the bubbles then will grow and meet into other bubbles, which then will pop open, make one great big bubble. And then as it's doing it, it's rewinding each of the bubbles so that by the time it's done, the two new DNA molecules are totally separated and rewound. Let's see if I can find that here. So here you have one here that will represent that. You've got a bubble where DNA polymerase has gone in, unwound the parent strands, and they are breaking apart the bonds between adenine, thymine, guanine, cytosine. They have to break those bonds. As they break the bonds, the two parental strands separate. New daughter strands are being added, and as they're being added, then they're being coiled up again. But you will have several of these bubbles all the way down the length of the DNA molecule. Think of it as you got 20 bubbles that are all working at the same time versus one bubble going from one end to the other. Which one's going to end up finishing first? Well, the 20 bubbles, right? Because you're doing 20 times as much, which is going to get the work done faster. Faster is better because the more the DNA molecule is exposed, the more chance it has of mutation. And that's not good. All right, so to minimize mutation, it wants to have that bubble open only a split fraction of a time. I mean, just boom, bam, up, down, replace, and close. Okay, and the only way you can do that is to do several of these all at the same time. All right. Moving on to slide 21 here. All right, so the replication fork, again, this is the replication fork, right? The replication fork moves along the parental DNA to continually unwind the parental strains and rewind the daughter strains. So again, if I showed you back at this one, you can see right here, these enzymes are unwinding. These are building and rewinding. So it's being rewound over here. So it unwinds, builds, rewinds. And it does that all at what's called the replication fork. All right, slide 22. DNA replication. It says the synthesis of complementary strand is catalyzed by polymerase, which is DNA or RNA polymerase. DNA polymerase, again, unwinds. Catalyze, think of catalyze as tearing things down, tearing things apart. Basically, if you think of them as just taking them apart, this is what DNA polymerase does. But it also helps to rewind everything. So DNA polymerase unwinds and rewinds. Okay, now this one down here where it says degradation of nucleic acid bonds. This is the bond between adenine, thymine, guanine, cytosine, is catalyzed by nucleases. Now the nucleases are like the endonuclease. It's a enzyme protein that will go in and break those bonds down. So polymerase unwinds, puts a strain on the bonds. The nuclease actually cuts the bonds. So it's kind of like you take the paper. If you don't have the paper stretched out, it's harder to break. DNA polymerase stretches that paper out. The nuclease comes in and is able to cut the bonds much more effectively. All right, so it's these two enzymes working together. Slide 22, this is the slide, of course, about endonuclease. This is where they does go in and breaks the bonds between adenine, thymine, guanine, and cytosine. All right, and then you have your last slide here, where you have the double helix has the ability to separate the two strands without disrupting covalent bonds. What it's talking about here, it does disrupt the bonds between the bases. This slide is talking about the bonds between phosphate and the sugar and the base itself. So the nucleotide stays intact. So this nucleotide bonds with this nucleotide via the bonds across the adenine, thymine, or guanine cytosine. Those bonds are broken, but the bonds between phosphate and the sugar and the base remain intact. The endonucleases do not affect those. The DNA polymerases do not affect those. Okay, so that is crucial. If it did affect those, the whole thing would just basically blow apart and things would be floating around everywhere. And to put it all back together would be a miracle if you could get it back together in the same order. All right. So that ends the notes.